Inside almost every single one of your cells, there's a molecule of just incredible complexity and beauty. It's the master script for the story of life, a code that connects you, me, and every living thing back to the very first organisms. We're talking about DNA, of course, and today we're going to unpack its biggest secrets. So, here's the plan. We'll start with the basics. What DNA actually is, and how its simple four-letter alphabet can write the entire code of life. Then we'll jump back in time to meet the brilliant scientist who cracked the code and figured out its structure. From there, we'll see how that discovery completely changed everything before looking ahead to the massive questions that are shaping our genetic future. Okay, so let's get right to it. What exactly is this incredible molecule that basically runs the show for all of life? Okay, the full name is a mouthful: deoxyribonucleic acid. The nucleic acid part just tells us it's found in the nucleus of our cells and has acidic properties. But really, what you need to know is that it's a polymer, a super long chain made of smaller repeating parts, all twisted up into that iconic double helix shape. And packed inside that famous spiral staircase are the instructions for, well, pretty much every living thing on the planet. Yeah, the blueprint analogy is seriously the best way to get it. Think of it as the ultimate instruction manual. It has every single piece of information needed not just to build an organism from scratch, but to keep it running and maintained for its entire life. All right, so if DNA is the blueprint, how does a cell, actually read it? How do you get from a string of chemicals to a living, breathing person? This is where we get into the code itself, how it's written, how it's copied, and most importantly, how it gets translated into the proteins that do all the work in our bodies. The code is built from these little units called nucleotides. You can think of them like individual letters in a massive, massive alphabet. Each one has three parts: a phosphate and a sugar, which form the long, twisting backbone of the DNA strand. But the really important part is the base. That's the letter that actually carries the information. And these letters, the bases, come in four flavors: adenine, guanine, cytosine, and thymine. We just call them A, G, C, and T. You can see they're sorted into two groups based on their chemical shape. The purines have a two-ring structure, and the pyrimidines have just a single ring. And this difference in their shape, it's absolutely critical for what comes next. And here it is, the golden rule, the most important part of the whole structure. The letters pair up, but they're very picky. A purine always has to pair with a pyrimidine. Specifically, adenine always pairs with thymine, and cytosine always pairs with guanine. A with T, C with G. That's it. It's that simple. But that rule is what makes everything else possible. It's the sequence of these pairs that makes up our genes. And that simple pairing rule, it's the genius behind how life copies itself. It's an incredible process. Before a cell can divide, it needs a perfect copy of its DNA. So an enzyme comes along and just zips the helix open, right down the middle. Because A can only pair with T and C with G, each of those separated strands becomes a perfect template for building a new one. The results? Two perfect, identical DNA molecules from the original one. It's beautiful. Now, what's really interesting is that in complex cells like ours, the DNA isn't all in one place. Most of it, our 46 chromosomes, is tucked away safely in the nucleus. But there's also a tiny bit of DNA found in our mitochondria, the little power plants in our cells. And what's so amazing about this mitochondrial DNA is that you only get it from your mom. It's a direct, unbroken line back through your maternal ancestors, which is an incredible tool for scientists. So, how did we figure all this out? It's an amazing story of discovery, full of brilliant minds, intense competition, and a few fascinating twists and turns. Let's unlock the secret of how we found the secret of life. Believe it or not, this story starts all the way back in 1869. That's when Friedrich Miescher first found this stuff in cells he called nuclein. For decades, scientists actually argued about whether our genetic material was made of protein or DNA. That debate was finally settled in 1952 by the Hershey-Chase experiment, which proved, definitively, that DNA was the stuff of genes. And that set the stage for the biggest discovery of all. But the real breakthrough, the key piece of evidence, came from an incredible scientist named Rosalind Franklin. She was a master of a technique called X-ray crystallography, and she took this picture, photo 51. It might not look like much at first, but to the trained eye, it was everything. That X pattern in the middle, that was the smoking gun. It screamed helix. So using Franklin's critical data, two other scientists, James Watson and Francis Crick, were able to build their famous model. And in their 1953 paper, they wrote this sentence, which has got to be one of the biggest understatements in the history of science. What they were saying was, hey, the way this thing is built, it tells you exactly how it can copy itself. It was the secret of life hiding in plain sight. Okay, so this discovery wasn't just some abstract thing for scientists in the lab. Unlocking this code has literally changed the world we live in ways that we experience every single day. I mean, just look at this list. The number of things we can do now is just mind-blowing. We're talking about everything from solving crimes with DNA evidence, to developing new medicines, to engineering crops that can survive a drought, all the way to mapping out the entire history of human migration and figuring out where we all came from. And this isn't just theoretical stuff. It has a real, tangible impact. A single hair at a crime scene can now put a criminal behind bars. In medicine, we're not just treating symptoms anymore. We can target the root, genetic cause of a disease. It's all possible because we're learning to read and work with a code of almost unimaginable scale. So, what kind of scale are we talking about? 3 billion base pairs. To put that in perspective, if you were to type out the genetic code from just one of your cells, it would fill a stack of books 200 feet high. And get this, that entire library of information is coiled up inside a space so small you can't even see it. It's absolutely mind-boggling. So that brings us to today, and to the future, because we've moved beyond just reading the code. Now we're starting to write it, and that's opening up a whole new world of incredible possibilities, but also some really, really tough ethical questions. Which leads us to probably one of the biggest questions our species has ever had to face. Just because we have the power to change the fundamental blueprint of life, does that mean we should? You've probably heard of CRISPR. It's a technology that lets us edit genes with incredible precision. On one hand, this can mean curing horrible genetic diseases like sickle cell anemia. But on the other hand, it opens up a whole can of worms, right? We're talking about designer babies and making changes that could affect the human gene pool forever. Then there's the issue of privacy. Who owns your genetic code? And now, with synthetic biology, we're even starting to build life from scratch, which makes us question what life even means. So when you really think about it, DNA is two things at the same time. It's this incredible historical document, a record of 4 billion years of trial and error in the story of life on Earth, but it's also the blueprint for everything that's yet to come. And that's what makes this moment in history so unique. For billions of years, evolution by natural selection was the only thing writing the story of life. But now, for the very first time, we're the ones holding the pen. The question that will define our future is, what story will we choose to write?